So, good morning or good afternoon or good evening. It depends on where we are in the world. And I wish you all welcome to this webinar organized by the Global Research Alliance Manure Management Network about measuring emissions of greenhouse gases and maybe a little bit ammonia from different compartments of the manure management chain and from livestock on grassland. So today we have three excellent scientists giving presentations about the emissions from the different compartments. We have Jesper Nørlem Kamp from Aarhus University, who will talk about how to measure from storages. We have Johannes Laubach, who is giving a presentation about mainly about emissions, I should say, from livestock on grassland. And then we have Paria Sefari from Wageningen, who is giving presentations on how to measure the emissions from livestock barns. And Johannes, sorry, I didn't say that you were from Agri Research, or maybe you have a new name now. We're all shifting very frequent. Sorry about that. So with these very few words, I will give the microphone or the screen to Jesper Nørlem Kamp. And Jesper, the screen is yours. Yeah.
Thank you. I just need to share. Yes, and I hope you all see my screen. Hello, everybody. Thanks for the introduction, Sven. I'll in this presentation talk about emissions from stored solid manure and land applied manure. And one common or one method that's been used in the I've using and have been using in the work that I'm presenting here is the inverse dispersion modeling methods, and this one is the backward Lagrangian stochastic model. So we'll dive right into it, and I think both Johannes and Maria later will also briefly touch on this method as well, that has been used for some of their measurements. So I'll just introduce it here, and you might hear another way to explain it, but I don't think that hurts to hear it in different ways. So this method provides specific source-specific emission rates, and the emission rate can be determined at different time intervals depending on the scope of your study. It is possible to use very complex geometry, as you for the source would need to know that very specifically how the shape of your source is. And you can either do that by accurately measuring the different sources and sensors, or by using GPS equipment to quantify where your sources and sensors are. Here is a picture showing see if I hope you can see. So we have a sensor here showing a concentration measurements. That's just a single point, and then from that point, based on the wind statistics that's measured here, shown by an ultrasonic anemometer measuring the wind, then with that wind statistics as input to the model goes backwards in time in different trajectories. That's the stochastic part of the model. So whenever you go backwards in time, you have touchdowns on ground, and whenever the touchdown is inside your source area, here shown brown for manure application. Whenever you have touchdown inside your source area, these touchdowns would end up contributing to the concentration to emission ratio that you have as output of your file, output of your model. And with a background corrected concentration measurements and the concentration to emission ratio, you end up with the specific emission rate from your source. So that's the basis for measurements I'm showing here. And the first one, sorry. So the first one is emission measurement from manure stockpiles. And the scope of this was to investigate both ammonia, methane, and nitrous oxide from a broiler litter stockpile, and to assess the impact of covering on the emissions. And this stockpile was with broiler litter, so that was from a commercial broiler litter house, where they used the litter material was peat moss mixed in with wood chips. And the clean litter material can be seen on the top right corner here. After the broilers were taken to the slaughterhouse, the litter was removed from the house and then directly put into the field in one stockpile, as can be seen in the middle photo here. It was 22 tons and initially 1.8 meter high, so a normal size used in Denmark. And then we aimed for, in this study, to use a single pile to have maximum coverage to see dynamics over time. So we had an uncovered period first, and then a covered period with plastic sheets. As you can see in the bottom picture here, the plastic held down with tire sites all over and sandbags on the pile, and then uncover the pile again for the last days. And in Denmark, it's mandatory to actually cover these all stockpiles immediately after they are built. So that's also why we wanted to investigate this, to see what the effect was when you did not cover on the first part of it, and then also see if there was some emission potential after removing cover at the end. We're fully aware that there are some drawbacks, and we don't have a case control in this scenario here. We've done other studies with other types of deep litter on manure that you can find if you're interested. So the way that we did was that we had a stockpile in the center here, and the geometry was mapped out with GPS, and then we had a sampling line measuring the line integrated concentration, and that sampling line was moved around the stockpile depending on the wind direction. So if wind direction was coming from south here, then we placed the sampling line just north of the pile, and then moved it around according to the different days where the wind was coming from different directions. Then we of course did filtering in order to only keep the most valid data. So we ended up removing 60% of all data, but we still had 859 half-hour intervals. So for the emissions, we can see these half-hour intervals. We can see them here in blue for ammonia at the top, methane in the middle, and nitrous oxide at the bottom. And the uncovered period here is marked in red, and then the covered period is here in the center. And it's clear that there are some trends. You can see that nitrous oxide, for example, at the bottom, increase in the uncovered period, and then when we cover it, decrease again. And then there is some bursts after the cover has been removed. And the same can be seen for ammonia, especially, that you have some emissions at the first uncovered period, and then when you remove the cover, there is also a large burst of ammonia being released, and then increased levels again. For methane, the picture is a bit more blurred, but the negative emissions here, both in nitrous oxide and in methane, they're not an indication of deposition. It's more an indication of the limitations of the measurement system, that we are close to the detection limits of the system. So in order not to overestimate the emissions, we include all emissions, also the negative ones, to make sure that we don't bias our results. But if we look at the total number, we see that 3 different periods. So the first uncovered, covered, and uncovered for ammonia, methane, and nitrous oxide. And it's clear that there is a high variation in the emissions, but that's also not only uncertainty in the measurements, but also shows how much natural variation there is in emission over time from these different compounds. Overall, we could see that the cover period has a large effect on ammonia and a smaller effect on methane, and taking the uncertainty or the variation into account, this is not significant in any way. And then for nitrous oxide, we could see that there were very low emissions from the covered period compared to the uncovered periods. And one reason why we are going for these large-scale manure piles is that there is a temperature development in these full-scale manure heaps. So this shows here the temperature at the outside air here at the bottom, and then we can see the top and the bottom, and then also the center or middle temperature. But there is quite high temperatures with 40 to 50 degrees inside the cover and inside the heap. So this is also a reason to be a bit careful to downscale or do lab scale experiments and extrapolate the results from that. And if you look at the climate impact on this using the GWP 100s values for the different ones, and then also includes that ammonia might not be a greenhouse gas, but there's also indirect contribution to nitrous oxide from ammonia volatilization at 1%, which means that you would end up here in the covered period having about 70 to 75% lower greenhouse gas emission in CO2 equivalents in the covered periods. So some conclusions on this study is that covering reduce ammonia and greenhouse gases in general, and one important outlook is also that in 22 there were more than 26 billion poultry in the world, and if all of them are kept in deep litter systems, there's a high potential to mitigate emissions if they are covered. And it's also a rather cost-effective method as it don't require any instrumentation or large machineries at the farm to cover in plastic tarp. So yeah, that's some of the conclusions. But of course, this is only one study, and in order to know more, it would be good to have more case control studies to show the real effects during the different phases, for example, and know more about the dynamics over time. So this was the first part, and then the next one is on ammonia emissions after field application of slurry. And as just mentioned, I know that ammonia is not a greenhouse gas, but there is the indirect contribution to nitrous oxides. And then there's also ammonia as being important for the local environments. So this here is, I'm presenting this because we did the method comparison to evaluate different measurement methods measured on the same plot. And we had methods that were both time averaged and spot samples in time, also absolute and relative. Emission factors were yielded from these, and then on-site, off-site measurements. And this was done in collaboration between Aarhus University, Wageningen University and Research, and then the Thünen Institute in Germany. And there are the different methods here, and these can be divided in different ways. So one way to do it is to look at the enclosure methods seen at the top here with wind tunnels and with flux chambers and the dragger tube methods, where you change the surface condition by putting a chamber on top of the surface. And then there is the micrometeorological methods, and that's basically BLS with different concentration measurements, so with impinges and alpha samples, along with cavity ring arm spectroscopy, but then also integrated horizontal flux with impinges. It can also be looked at that we have some measurements that measure in high time resolution with the online cavity ring arm spectroscopy and concentration measurements, some with spot sampling time being the dragger tube, and then those that integrate over time with impinges and the alpha samplers. And we did 2 trials in this year, one in Denmark with wind tunnels, BLS with cavity ring arm spectroscopy and alpha samplers, and then also DTM. And then we did one in the Netherlands, where we had wind tunnels and BLS with cavity ring arm spectroscopy and impinges, but also IHF and flux chambers. And going straight to the result here, then we can see that we have in Denmark, we applied with trailing hose during the summer, and in the Netherlands during winter with injection. And just keep in mind that the scale is not the same in the two graphs here. We have high emissions in Denmark during summer and lower during winter in the Netherlands. And that's, of course, both due to the application technique with trailing hose giving higher emissions than injection, and also the season with winter giving lower emissions. So we can see the different dynamics here over time, but if we then go to the cumulative loss here as loss of percent applied TAN, so how much total ammonical nitrogen has been applied, that's the measure here. Then we can see that quite a lot of difference between the different methods, and it's very clear first that the first 24 hours after application is the most important for the total losses, but we can also see some different trends here. And with the enclosure methods with wind tunnels, for example, we can see that over here we had 3 different air exchange rates. So how much air was pulled through each of the chamber, that ended up giving quite different results in the total loss of ammonia. And the same can be seen here, where we had the flux chambers offsite compared to wind tunnels. That was also different. We could also see that for when using the IHF, where you needed to fit the wind profile, we couldn't see any emissions after 70 hours, while if we used the BLS method to directly on the impinge concentration measurements also used in IHF, we could still see an increase in emission after 70 hours. And then there are also some different trends here. We can see that the DTM giving only spot samples in time, and then interpolating between measuring times, that gave quite low emissions overall compared to all other methods. We can also see that BLS with alpha sampler in this case here gave quite different or quite lower emissions than the BLS with cavity ring arm spectroscopy in the red line here. And opposite was the case in another study on another measurement in the Netherlands, where we saw that BLS with impinges gave higher emissions than the BLS with cavity ring arm spectroscopy, so in the red lines here. Bit of a funny thing, in the Netherlands we had a rainfall of 4.5 millimeters over 3 hours, where you can see that the blue wind tunnel here that's enclosed, that had no effect on that with this rainfall, but we can see a tendency for the BLS measurements to plateau out after this rainfall. So that's also a reason why these enclosure methods should only be used for comparisons and not to give direct emission factors. So some conclusions from this study here is that of course, there's difference in emission depending on season application methods, and also that you can see the different flux dynamics with the high time resolution measurements. And one specific importance to the application of slurry is also that all the explanatory variables, they're very important and should be recorded as much as possible. But I think the key output from this, and what can also be seen in some of the modeling work, is that the measurement methods chosen, they affect what the actual reported emissions they are. So yeah, I think that's a key element to this study, that even though we measured on the same source with all the different methods done by esteemed researchers within for each of the different methods, we still saw a difference with the different measurement methods, and that should also be taken into account as an uncertainty when you compare different studies to each other. And the last part here was on ammonia. So I just have a few points for those if you haven't measured on ammonia or in general just on your system. So concentration measurements on ammonia can be very tricky. And one thing is the response time being rather long. So this can especially be important if you have a multi-position system where you change between different positions with valves or something like that, that you risk of bias your results to a high degree if you have insufficient measurement time, depending on which method you're using. You can see that there's almost an hour here, and one of the systems didn't reach the set point after a step change in concentration. And this is also, of course, nitrous oxide and methane is not sticky in the same way, but I think it's important to validate your system when you are measuring with sampling lines to see what the response time is in order to ensure that you actually measure the true contribution from the source that you want. Also, interference, when you measure concentrations, you risk having false signals from non-target gases, and a study that we did show that you actually have false ammonia signal with one instrument, rather high from some of the VOCs that you can often see in an agricultural environment. And the last point I have is on the background level. This is shown here for ammonia, that fluctuates a bit more than some of the other, but if you normally have diurnal variation of the ammonia background level. So if you have a low emitting source, then it's important to know the sensitivity of your concentration measurements. And this is also shown here that when you look at the background level done with different methods, then you might not end up seeing these small changes. So when you measure here very large concentration at first, small differences of a few micrograms per cubic meter is not that important. But when you get to later stage where you are closer to the detection limits, very important to know the limitations of your system if the sensitivity is not high enough. And yeah, that leads to the conclusion of my presentation here. And you are you're welcome to contact me if you have any questions or anything else. And at Aarhus University in our section and group, we are always ready for collaboration. So please reach out if you have any questions or anything else.
Thank you very much, Jesper, for this introduction to using to use different technologies, but very much to use the integrated, to use the backwards Lagrangian stochastical method. And I know you're using very, very fine and very expensive instruments. So my question now would be that, what do you think if I am in an area where we do not have maybe we are not even having power, would we then be able to carry out a meaningful study where we are using syringes to collect gas samples for methane and nitrous oxide, and we maybe have an anemometer that are that are not, with a battery and not fairly precise, but not very precise. Would that be an option?
So if you don't have, for example, an ultrasonic to quantify the wind field very accurately, then you'd need to measure the concentration, the wind in different heights so that you can actually make a profile. But it still needs to be a rather high time resolution. And then, of course, you can sample, but the issue is that you need to have stationarity. So the conditions needs to be stationary in the time interval where you measure. So taking out samples would only give a spot sample in time. So I think it can be tricky also with the passive samplers measuring over a very long time. I think it can be a bit tricky to actually use the model over, say, half a day, because the conditions would change, the wind direction would change, so the influence of your source area would also affect the outcome. So it's tricky also for
Yes.
alpha samplers shown here for ammonia or impinges. If you average over 6, 12, 24 hours, then you still have the issues of non-stationarity conditions, changing wind conditions, and things like that. So I think it's a bit tricky to use it in that sense. So yeah, you should ideally have concentration measurements that's taken continuously or sampled over a shorter time, and then you can, in these shorter time intervals, say for example up to an hour, then you can use the BLS methods with that. But
But
if
could
you
I put
don't,
it like, but maybe if we did put your gas sampling devices in the center of your plot, but then one maybe could just use the integrated horizontal flux technique or instead of the BLS.
yes, if you have of course, if you have a circular source and you are at the center, then you eliminate these factors regarding wind direction, but you still risk having very different conditions for the model. And then when you average the model input, then you get one average out. So I think it's a bit uncertain how to deal with these longer time intervals.
Oh, but I was mentioning a syringe, so that's actually within a very short period.
Yeah, and that's another point. If you take out samples, then you give an instantaneous concentration. So that can be done, but then you risk that you had a minute before or 2 minutes later having a burst of
Yeah,
methane, for example.
yeah,
So that's also gives some uncertainty. But I think that's no different than, for example, what you're doing with nitrous oxide and static chambers, where you place them on top and measure on shorter time. So I think you deal with that with taking a lot of measurements to know how the variation is over time, and then when you've quantified that, then you can do a more program
yeah.
set up
Thank you.
based
So
based on that.
it's just to make it more simple and give more people an option to carry out. That's why I'm asking. So thank you very much. I don't know if there are other questions. I think there's one.
So for
Uh,
has there
it's
been
it's
has
a
there been any
it's
study on effect of plastic covers or variation in cover types on emission? Um, I know that in our group there we've made a study where we looked at the same plastic, but how well it was placed on top. So if it was a bit sloppy or there are holes in the plastic or not covering fully, we have a study on that, and we saw that even a sloppy cover or a cover with some holes is better, especially still has a high effect for ammonia, and for methane in that study we could not see any effect at all. So yes, it has been investigated in that sense, but different types of cover we have not looked at that. I'm not familiar if there are any other studies that has done that.
So Jesper, I think there are no more questions. People can, you have said they're most welcome to contact you later, so that's great. So thank you very much for your fine presentation. It was a very good introduction to the techniques. And now I will give the word to Johannes. And Johannes, maybe you'll just give a short introduction to yourself. Yeah.
Um, Can you see my screen and presenter mode?
You're on calendar.
It is not showing it.
It's fine, Johannes. We can see it now.
You can
But
see it
maybe you
now.
have to, yeah, but
Okay.
you should show it in presentation.
Johannes, in the top, if you say display settings, then you can see SWOT. Now we
yeah.
see
There
the
we are.
presentation.
All good now?
Yeah, it's fine.
Great, excellent. Yeah, so I'm Johannes Laubach. Thank you for the opportunity to present about micrometeorological methods to measure greenhouse gas emissions from livestock. Briefly about me, I'm based in Lincoln in the South Island of New Zealand, and I work for what used to be a Crown Research Institute called Manaki Whenua Land Care Research for some 30 years or so. And as of July this year, we have been merged with 3 other Crown Research Institutes and are now called the Bioeconomy Science Institute. I've been in the field of measuring greenhouse gas emissions for a long time, and I would like to, where's the forward now? Start with the motivations for doing such measurements. That begins with need to report greenhouse gas emissions in national inventories. And because there's quite a variety of livestock types around the world, different feed sources for them, and different management systems, there's quite some variation of how the emissions could turn out, and to quantify that we just need more measurements of these different systems. Then there is an urgent need to develop mitigation approaches, and once suitable candidates are identified, it needs to be validated that these mitigation methods work in real-life farm systems and that they don't lead to unintended trade-offs. And then the next step, once mitigation methods get adopted by farmers and used regularly, then again there's a need to account for them in inventories, and so the average effectiveness needs to be quantified again. And then even more detailed would be if in the future we move to a detailed accounting system on farm level. To question now is first, what do I mean by micrometeorological methods? I try to put it in one sentence. These are methods that combine measurements of gas concentrations or mole fractions in the lowest atmospheric layer near the ground, with information about the transport and mixing processes in these layers, so that one can calculate gas exchange rates from that. There are a few methods that do that. These include the eddy covariance method, gradient methods, mass balance, inverse dispersion, some tracer ratio methods. I will not go into all of them, just a few. What they have in common is that they work with this situation here. An emission occurs from some sources like animals. The emitted trace gas is then transported in the horizontal by the wind and at the same time mixed in the vertical by turbulence, which is rapid during the day and in calm conditions slowing down, which often happens at night. One approach to measure in this situation is the eddy covariance method. For that, one needs to measure both the gas concentration and the vertical wind very fast, several times a second, and ideally in the same place, so in practice close together. And then the covariance of the time signals for concentration and for wind gives the vertical flux at this point. And this flux, the eddy flux, is equal to the emission rate in the upwind area if the source distribution is homogeneous for this area. If it's not homogeneous, it's unevenly distributed, then one needs to consider the so-called footprint. So that's extra theory to express how different areas upwind contribute with different probabilities to the flux measured at this point. Now, these measurements require a fast concentration instrument. There are not many options available and they tend to be expensive. And so an alternative, if you can't afford that, would be to use an analyzer that is slower. It needs to be precise and it needs to measure at two different heights then. So you get an accurate difference in concentration between these two heights. It's called the gradient. The gradient is proportional to the vertical flux. And so with the same assumptions about homogeneity of the sources, you then again get an emission rate calculated. So both the eddy covariance and gradient methods are suitable for sources that are spread out and reasonably homogeneous. So they are good for nitrous oxide emissions from soil. They are good for CO2 exchange of vegetation. And in fact, there are hundreds of flux towers around the world measuring CO2 exchange over different ecosystems. The method can also be used, as sketched on the right-hand side here, to compare two systems. If you measure at the boundary of the two with one system, and dependent on wind direction, you get a signal either from system 1 or from system 2. And because you do it continuously over time, you build enough of a data set that you get both systems represented well in their emissions and you can compare them. However, when you're actually dealing with livestock, that means animals that can move around, that constitutes a practical challenge because they move often in ways that are not evenly spread, they can cluster together, and this all causes great uncertainty in the flux emission relationship that is necessary to use. And the second challenge with animals is particularly in New Zealand, where rotational grazing is used on dairy farms. The cows need to move to a new paddock every day. Then you either need to move your instruments to follow them, or you are relying on very narrow wind direction sectors if you try measuring a few paddocks in sequence. And so for these practical challenges, I would not in general recommend these methods for grazing livestock. However, where they are good is in feedlots, because that's a pretty homogeneous source and a large source, and so it has been successfully shown that methane emissions from feedlots can be measured, and you can resolve diurnal causes of emissions, and that shows an advantage of this method, the high temporal resolution, which you cannot get with a number of animal-based methods. So, but we have more other methods in our toolkit, and an important one is the inverse dispersion model method. And Jesper has already explained this in much detail in his talk. So very briefly, it's for the situation here that the animals are in a fenced area, so we know where they are. We measure the gas concentration upwind of them and downwind of them. We still measure wind and turbulence, and use a flow model, which then calculates backward trajectories of air parcels to map out where they came from. And it does that many thousands of times. It's a simulation with a random element for the turbulence, and so it creates a probability map of where these air parcels came from, and those inside the fenced area contribute to the emissions, those outside don't, and that way the emission rate can be calculated. A straightforward application of this method is this one here, where in Africa we do not have a lot of measurements from livestock. So these colleagues said, well, in Kenya, the herders have this old practice that overnight they put the animals into an enclosure called a boma to protect them from predators. And so while the animals are in there, it's very easy to put up a methane laser, measure upwind, measure downwind, and get good capture of the methane plume, and can from that calculate the emissions, and then also the emissions per live weight of the cows, which is an emission factor. And so they were able to get some real data points with uncertainty for this emission factor, and could compare that to a few studies where these emission factors had been modeled with good agreement in this case. So this is a good application of the method to give real data for understudied systems. Now, apart from having emissions for groups of animals, Jesper already showed that you can apply the method to manure treatment systems, piles, ponds. You can also use it around animal houses. The method can use concentrations measured at a point or integrated over a line. And one downwind, one upwind is enough. But if you measure at more locations, it will improve the accuracy. The practical challenge, one particular, if you want to compare two systems, is you have to make sure that the emission plumes from these two systems do not overlap. And so you have to separate the two systems either in space, or you have to separate them in time, which means you measure at different times. The other challenge of the rotational grazing, of course, remains if the animals move often, your instruments have to move as well. Now, one very neat application of this method to compare two systems is this one here. It was to evaluate feed additive 3-nitrooxypropanol. And these colleagues used two pens in a feedlot, one where the cows were given the feed additive, the other one where they were not given it. You see there's the distance left in between so that the emission plumes don't interfere with each other. Where these black bars are, that's where this research group put their methane lasers to measure the concentrations, and they knew that the dominant direction would be from the south, so they would get good data coverage. And with this setup, they could show that the overall emissions reduction over a continuous period of 4 months was about 70%. And the mitigation effect decreased over time, which is an important result for evaluating a mitigation method like that. Now, 70% is of course a large effect, so it's relatively easy to measure. Can we do better? Well, the answer is we certainly can. Now, at our own research group here, we have opted not to use a laser system, but instead want to use high accuracy gas analyzers, which work with closed cells. And so we have to bring the air into these analyzers, and we are using 40-meter long pipes with holes in them to suck the air into the analyzer. And we have designed the system so that we can investigate emissions from groups of about 20 to 30 cows, like shown on the right-hand side here. The red lines are positions of the pipes around these two areas. The blue area was where the cows grazed first. Then they went for milking, came back and grazed the gray area. And so we could cover both periods with the same setup without having to move it. And got our emissions data. Now, this is an ongoing project comparing two different grazing managements, so I can't show the results of that yet. But some years ago, we did already the groundwork for this 4-pipe system, and we showed that we were able to detect a difference of 10% in group emissions. Which is quite a bit more challenging than detecting 70% difference. Now, there are colleagues in Canada who went a bit of different path in that direction. They have an optical system which, by using mirrors, they can multiply the path. And they decided the best setup would be to have long, narrow grazing strips, because that optimized in their setup the data coverage for all wind directions. And if they use the setup in multiple campaigns, then they were able to show that they detected a 6.3% emission difference between two feed treatments. So using the statistical power of multiple campaigns, this is the lowest confirmed effect number that I've seen to be achieved with micromet measurements. Yeah, so much about few specific examples. Back to micromet methods in general, they have a few disadvantages. The main one being in treatment control studies, you are restricted to just 1 or 2 replicates because more is just too expensive or difficult to handle usually. And you cannot trace the emissions back to individual animals. So what you're getting is group emissions. But there are many advantages for these methods. An important one is the measurements do not affect the animal behavior. They don't interfere with the farm management. So they are non-intrusive, very important. These methods integrate over a whole field or paddock. They can also be used around houses, ponds, or treatment systems. And if you automate the data acquisition, they can run for long periods, and then they can resolve seasonal patterns. They can also resolve dial patterns because of the high time resolution. And that is useful because it provides detailed information for process models which can then explore different scenarios and quantify associated uncertainties. It can make recommendations how to better manage the grazing situation. So to conclude, there is a variety of micromet methods, and they can be tailored to suit your problem, suit the source configuration and your measurement goals. It's not one approach fits everything. And there are demonstrated uses that can underpin and improve national inventories. And the method can also be tailored to resolve management differences of order 10 to 15%. And that is very useful for management practices that you cannot investigate with chambers, for example. What remains a challenge is the verification of emission accounting systems. That's still a bit in the future for reasons of cost and numbers of just sheer numbers of farms that would need to be done at. And just a final bit of self-promotion. A lot of the facts and ideas that I presented here you can read in a review paper that we published last year in Agriculture and Forest Meteorology. Thank you very much.
Thank you very much, Johannes, for a good introduction to these methods following up on Jesper's. You were saying that you can run, I'll just ask a few questions and then more will come, hopefully. You can run this the measurements for a long period. Would that demand a lot of labor, and is it to maintain your instrumentation?
It depends. First of all, which measurement method it is, and then of course, if you want to run it for a long time, you want it to stay in the same place. So for rotational grazing, it's not a great not a great approach there. But that is challenging anyway. So following moving cattle over a long time, that's a very hard one. But for like the feedlot situation, for example, you could just run the instruments and occasionally check that the data are good, and so that would not be too labor intensive. You probably need a bit of information, of course, whether there are changes in the usage of the pins around the feedlot, for example. So, I mean, with any of these, of course, you collecting your micromet data is one thing, but you also need to collect the management information so that you can interpret what you are measuring there.
Thank you. There are some questions now. Can you read them, or?
I don't see them yet. I've stopped sharing, and now I can. Did the experiments look at variations in seasons and the feeding regimes of the animals? Not sure which experiments you refer to here, but if it depends, of course, if your experiment is set up long-term to distinguish between seasons, or if you do multiple campaigns at different seasons, then yes, you can distinguish that. And also with the feeding regimes. I don't know if that has answered the question well. The next question, do you recommend this method to the sub-Sahara African countries where the livestock production system is on natural grazing and rangelands? Well, if it's about just gathering first data, then I think I would recommend reasonably short campaigns, and yeah, find a location, and collaborators, farmers or herders, where you can set that up for. And yes, you would need a measurement system that's reasonably low power. I think some of these methane laser systems can do that. They can run on batteries or solar. So yes, that is possible. The third question here: is it possible to use the method to measure emissions from the animal yard? Yes, in general. One general assumption for micromet that I haven't stressed, maybe should have, is all these models are in general for flat terrain. So you need to look a bit into that, because if you have sloping terrain or hilly terrain, then the flow situation is just more complicated. And so find a site that's reasonably flat. And then if you have a yard in there or a house in there and you set up with sort of enough distance around that, that there are no direct obstacle effects, then yes, you can apply these methods there.
I think there.
Right, quite a few more. Do we have time to go through all of them? Or
I think let's take 4 more. I'm so sorry. But yeah.
Is there any work using different micromet methods in a single experiment to see which one is robust? Is there a golden standard? I wouldn't say there's a golden standard. I've been involved in a few comparison experiments, and will be again next year. I'd say one good comparison is of these methods to tracer ratio methods. By that I mean, if you release a gas at a known rate in sort of the same locations as the animals or whatever your emission source under investigation, and then you measure the emission plume of both gases, the one that you release at a known rate and the one that you want to measure, then this tracer ratio method bypasses all the assumptions of the micrometeorology, because the only assumption is that both gases are transported by the same turbulent structures. So that is a kind of a good standard, if you wish, to compare to, which cannot be applied long term because you simply cannot afford to release a gas for more than a few hours or a few days. What do you think of deposition issue of gas, e.g. ammonia, on the path from source to sensor? So if I understand it right, then this is about the fact that ammonia may be emitted by a source, but then some of it gets returned and sticks to the surface before it actually reaches your concentration measurement. That's true, of course. What you in effect measure is a net effect. And it depends on the circumstances of your measurement situation, whether you need to model something about that or not. Are the results of these methods compared with standard options like chamber and green feed? I don't like the word standard in there. But I'm involved in a group of scientists from New Zealand who are planning next year to compare a few methods which includes the green feed. And so it will be interesting to have the green feed and the micromet applied to the same group of cows.
Johannes, I think maybe this is the last question. Is that okay?
Yes, that's fine with me.
Yeah, I'm so sorry, but
I'm happy if I get email record of these questions. I'm happy to reply to them by email if that's an option. Yeah.
And maybe the question with ammonia deposition can be quite interesting, actually, if you have your sensors far a bit distant from your source. But that's an ammonia person speaking. So thank you very much, Johannes. It was great to hear you and to see you. And I think we learned a lot, as we did from Jesper. So, and I'm sure you have given people, you said people can contact you and learn more about how to use the method correctly and where it can be used. So with these words, we will move to Pari Asifetpari, who will give a presentation about the emissions from barns and how we can measure that. So the word is yours, and you might present yourself first, please. Kind. Yeah. Thank you.
Thank you, Sven. Hello, everyone. My name is Pari Asifetpari. I'm a researcher at Wageningen Livestock Research, part of Wageningen University and Research. I'm very happy to have this opportunity to present in this webinar about micrometeorological methods. Indeed, in my work, we focus on more than that. So it's about emissions from livestock systems, particularly the source-oriented mitigation measures for animal houses. And my focus is mainly on pig housing systems, manure management, and measurement methods and modeling of greenhouse and ammonia emissions. Today, I'm presenting some insights from our recent work at Wageningen on evaluation of emissions from pig housing systems with an outdoor area for pigs, and we focus on application of the micrometeorological methods using the backward Lagrangian and stochastic models. It's a part of an ongoing research in which I, and together with my colleagues, are active in, and I'm pleased to be part of this session and look forward to your questions and discussions afterwards. I hope you see my screen with first slide, so I will move on.
We see it.
Yeah. In the Netherlands, of course, agriculture, such as the other parts of the world, is responsible for around 50% of the total carbon dioxide emissions. And within the livestock system, pig farming is also contributing to that with around 15%. To methane emissions, up to 20% can also be assumed. But agriculture is also a source of ammonia emissions, and within agricultural sector, pig farming alone is also contributing to that. And showing these figures together with the plots on the right side, it can be concluded that manure management from the livestock housing, of course, is very important. So in the national greenhouse and ammonia emission profile. So in order to get insight into the current situation of the emissions from pig houses, as well as also assessment of the future measures, measuring and monitoring of emissions are actually crucial. With that, we know that there are still significant data gaps regarding emissions from naturally ventilated pig farms. with an outdoor, and these systems are actually increasingly becoming common in different parts of the world, including the Netherlands, but poorly represented in the emission databases. So at present, there are no comprehensive overview exists of their annual emission factors, and no standardized measurement protocol also has been developed for these farm types. So as a result, there are no official emission factors, let's say, established yet to be used in national inventories. So these systems are actually, on the other side, are expected to have low emissions due to the application of source-oriented measures, and because of the open structure and the outdoor access. So that makes it important to quantify and validate their performance with reliable measurement methods. Based on these knowledge gaps that we just discussed, also in other presentations, we also in this research focused on three main questions. So the first one is how we can measure emissions from naturally ventilated pig farms. What is the quantification method and how these methods can be actually standardized. Secondly, we would also like to show the influence of the weather conditions and animal behavior on emissions, and we want to understand that how this variability actually influence interpreting the measurement results. And finally, to test a method or multiple methods that can be hopefully be aligned at the end with respect to the results. The farms that we selected with these outdoor runs actually are pig farms in the Netherlands with an animal welfare friendly concept. They include two parts, the naturally ventilated barn and a roofed area, partly roofed area outside with an outdoor access for the pigs, where 95% of the manure production is occurring there. So we expect that most of the emissions are produced from the outside. The barns inside, the pens inside can be adjusted. And because this is a stress-free housing, it means pigs will remain in the same barn and pens during the whole growing period, and the pen sizes can be adjusted, of course. There are 2 production cycles per year. By that, it is also tried to control the optimal behavior of the animals and the indoor climate. Outside of the barn, there is a direct manure separation and frequent manure removal system under the slatted floor, which are metal. And manure is separately stored in urine and feces fractions. For this specific farm concept, we have decided to measure emissions with 2 methods. The first one is at barn level using the micrometeorological method. And the second method is at pen level, which is an ongoing research of this year and next year, in which we measure ammonia emissions from the surface using the so-called chamber methods. And these measurements, together with process-based models and vision techniques to detect the urine puddles on the floor and situation of the animal behavior, we would like to model ammonia emissions and then compare it with the first method. Already we have heard a lot about the micrometeorological method, so I would like to keep it also short here. In this approach, we work backwards from a point where the concentrations are measured of different gases can be also, and to estimate where and how much gas must have been emitted at the source area. So we follow the trajectories back to the source. In this method, we apply the BLF model R package in R, but we also compared it with wind track because we started with wind tracks, but we all know that they are the same underlying principles. So in short, the BLS model simulated the turbulence characteristics in time by the dispersion model. And this is actually the principle where the BLS model can calculate the CQ ratio. And then we measure concentrations upwind and downwind. And the difference of that divided by this simulated ratio can give an indication on the emission rate. To approach this method, we used a setup in which a gas analyzer, multi-purpose gas analyzer was used from Picaro. Measurement intervals for different lines were background and also downwind of the source were 10 minutes. However, looking at the pattern of the measured concentration, we realized that we should actually select the last 2 minutes. We also used the 3D sonic anemometer to capture the wind characteristics, and also temperature and humidity were measured and logged. And you can see below the main items of this setup, and later I will show performed and how this setup were formed. In order to actually comply with the literature, we know that we should go far from the source, so 10 times the height of the source was selected at 80 meter, 75 to 80 meter difference in different farms. But in order to check how this will be actually affected if we come closer, because of also dilution of the concentration. So we came also to 40 meter, and then we set another line, both at 1.5 to 2 meter height. We did these measurements during 5 periods of around 1 week, more or less, and with different animal categories involved in each. At the end, we also did some recovery tests of 1 hour, release of nitrous oxide. At case farm 2, we had a slightly different situation because there were 2 of these barns parallel to each other with the outer run in the middle, as you see in the photo with the pink pig. And there was some other actually limitation to set up the line, but we could still set up the line parallel to the length of the barn. And we had less number of measurement rounds, but still continues over several days. At farm 3, we had some more challenges. It was difficult to set up 2 lines parallel to the length of the barn, according to the prevailing wind, which is mostly southwest in the Netherlands. And that direction, we had also a row of not very intense trees, but they were still there, and roads, etc. So we decided in the second setup on the right side of the slide to make the second line parallel to the length to see how this will differ. To go into the results, I will show only the calculated emissions from methane and also ammonia in this slide. On farm 1, this was actually a farm with a relatively optimal situation. And as we see here, the lines at 40 and 80 meter are quite comparable in terms of calculated emissions. So it's consistent through different measurements. But we see that, okay, there are still some variations, especially for ammonia emissions, which we should figure it out, which factors are affecting on that. For methane emissions, of course, there, you can see also negative if you look at the different scales of the plots. Negative values were measured, but these values had to be taken into account since it can be related to the limitation of the setup or deviation of the downwind conditions from background concentrations. So these were also included in the total emission calculation. Based on these results, so we can also extrapolate that to the to a normal emission factor unit which is actually in our national inventory, and any kilogram methane or ammonia per year per animal place. The most important conclusions, comparison of the emission levels at two different distances were in good agreement. So probably it's good to focus in future research on measuring closer to the source, considering the turbulence conditions, of course. Based on the recovery ratio from the nitrous oxide, we found that 72% of the emissions were captured in an optimal situation, which is good, but still relatively low. Which means that the setup can still be optimized. However, on farm 3, we saw a lot of inconsistency, but also low recovery because of the suboptimal setup there, and difficulty to set up the lines facing the outdoor air run for the pigs. Furthermore, there were consistent results between WinTrax and the R package. Data were not shown here actually. What we have observed up to now is that this method could also confirm the model predictions or expectations from this system in terms of emission reduction. So we had certain expectations from this specific farm, and with this method, we compared to the standard emission factors for a pig farm, we could get satisfactory results. Some concluding messages and future prospects about the micrometeorological method application in livestock housing. I should say that many livestock houses are actually associated with aerodynamic complexity, so we should consider that, and that complicates the application of the micrometeorological method. Measurements far enough from the downwind of the source are necessary. However, there is this limit to dilute bloom samples, which leads to uncertainties of the measurements, and we should take that into account in future research or to repeat closer to the barn. For deposition gases, like depositing gases, for example, like ammonia, deposition to the surfaces such as soil, vegetation, or building materials and obstacles can also lead to underestimation of emissions if not corrected for that. We also recommend to do some sensitivity analysis of the key BLS model parameters, as well as uncertainty on the model outputs. Of course, further validation of this method remains important, both for ammonia, but also for other gases like methane. By that, I would like to thank our team involved in this research, but also the pig farmers and also the province of Gelderland, who has financed this project. We would be happy if you have any questions or any collaborations can be established in future to contact us. You know where to find us. And thank you for your attention.
Thank you very much, Paria, for a marvelous presentation and for giving us new data about how emission can be measured from naturally ventilated barns. I have a few questions for a start. That's my benefit. It's one, as far as I could see, you did have ammonia deposition. You measured lower emissions at far away than close to the barn. How much? Do you have an idea of how much that would be?
You mean the position?
Yes, the deposition, yes.
Yeah, that has not been quantified yet in this research, because that's a complicated addition to the current model. We need to accept that there is an underestimation at this moment because it has not been corrected. But but that should be discussed actually about
Yeah.
the results of such applications.
So
Yeah.
you'll get that one, and then when you went closer to the barn, you said you would get, as far as I could see, the methane emission estimates were similar for 40 meters and 75 meters. Is that correct?
Yes. Yeah. For methane, we could see more consistent results at the second line closer to the source. The same is true for ammonia, but ammonia, the variation was a bit higher, especially for some rounds where fatteners were involved, and that's the or during the summer period. So we still need to really focus on what has caused this variation, and yeah, to also look into more into the relation between wind turbulences of factors and other factors like yeah, the season and the status of the manure system and the animals themselves.
And then maybe my last question, I'm sorry, but it's when you were measuring the tracer N2O and you measured at 75 meters and 40 meters, would you then could you then see a better recovery at 40 meters than 72, or didn't it?
Indeed, there were consistent results between the two lines. So that's why we, and because the emissions were consistent, so we took the average. Here we showed the average of both lines, but yeah, in terms of the recovery, there were no significant differences in the recovery ratio compared to what we measured there with the target gas.
Okay.
So,
thank you very much. I
yeah,
will give
okay,
the
welcome.
audience a chance to ask you questions. So if you look at the
Yes.
question down there.
I think I can look into the questions from
then answer,
8 o'clock. Yeah. The first one, I don't think it's at exactly at 8 o'clock. I don't think that's a question relevant to our study, because it's have you measured the soil nutrients such as nitrate and nitrogens to understand the gas emissions with those parameters. Indeed, that was not the goal of our study. So probably this question is more related to presentation of Johannes. The next one was from West Africa, I believe. Oh yeah, okay. That's thank you very much, Joel, for your introduction. Next question: did you observe any correlation between the diurnal patterns of methane and ammonia? Yeah, indeed. So that's actually related to the wind characteristics, we believe. So that the diurnal patterns were seen for ammonia, but also for methane and nitrous oxide gas, because we also measured that. Yeah. The last question, considering the complex terrain and the wind obstacles, as well as the extremely short measurement periods, that's 1 week, what were the turbulence stress statistics and data retention at each site? What were the filtering criteria that you used at each site? Thank you. That's a very good question. Indeed, I didn't go through the criteria, but we tried to follow the previous studies in terms of the criteria, the upper and the lower boundary of the BLS model parameters to filter the data. We also filtered the data, obviously, based on the predominant wind direction on those periods that we did the measurements. And as I told you, that the last 2 minutes of the gas concentration data were directly used in the measurement. So these were the most important criteria that we took into account for the measurements. But because the measurements were short, relatively shortly, yeah, the data retention at each site was actually different. We have also we have the statistics about the number of data and the retention of the data, of course, per measurement now, but these were not shown here. So probably we can communicate in future, because that's also a very important point, that how much data we retain and how that's representative for the measurement period is. But in general, I should say that it was a satisfactory situation that we could use those data sets from those measurement routes in the calculations.
Oh,
I think that was the final question. Is that
oh,
right?
there's one offering that we could, and that it would be great to carry out this testing of the methods in. Here we are invited to. You are invited to Nigeria. So because I just follow up on this, there are very few measurements on gas emissions in South Sahel Africa. So that would be great if that could be set up.
Of course, yes.
So we have 2 more questions, and then I think we have to say, once more, good morning and good night, and finish the webinar.
Should I look at the chat?
Yeah, is it possible if we use high resolution remote sensing data to detect and map methane?
Oh, yeah. Is it possible if you use remote sensing data to detect and map methane? Your recommendation. Yeah, indeed. I
for
think
Johannes' presentation earlier.
Okay, so it's from Johannes.
probably,
Sorry.
yeah.
So
Please, Johannes, go ahead. Yeah.
So Johannes will have the final answer to questions.
Yeah, I have actually been involved in a project in that was using the methane satellite, which unfortunately the satellite failed a few weeks ago, so there won't be any more data coming from it. But it's using remote sensing data to map methane is one thing you can do, but the attribution of that methane back to sources is a very complicated problem then, which was meant to be tried in this project with inverse modeling. And there are resolution challenges with it. So I think it's worth attempting more in that direction, but we are not there yet. I don't know if the question maybe also includes sensing from closer by using drones or something. But it always comes to the question, what do you need to do to get from a concentration field that you have observed back to the emission sources and the attribution of emission strength then. So it's complicated.
And then that would be the final questions and presentation. Thank you very much, Paria, Johannes, and Jesper, for giving very good, enlightening presentations. And I think it will make most of us curious and interested in using the method, and also to use it correctly, and maybe using, making it more simple. Somewhere in the world, we cannot use very complex, difficult, and expensive to use equipment. So that would be maybe some of the questions sent to you. So I hope people will use the opportunity to ask you questions on the email or phone you or visit you. And so that this is a starting point for some of us to use the technology and use it correctly. So thank you very much for your presentations. We appreciate much that you spent your time on this. Thank you. And then goodbye also to all the audience having listening to you. So goodbye.
Thank
Thank
you
you.
and goodbye.
Bye.